it is my great pleasure to introduce you Dr. Joseph Baur from the Department of Physiology, who's going to be talking about NAD metabolism in mammalian mitochondria.
So I want to start by thanking Claire for that introduction and Josh and all of you for being here and giving us this opportunity to tell you about some of the recent work going on in my lab. So I want to start with the disclaimer that my lab doesn't work on the eye. We're broadly interested in aging and in different interventions that might be useful to slow down age-related diseases. So I made this slide a while ago, and I realized putting this together that I actually got one prediction right, but have to apologize to Keith Richards, since he is actually doing much better than expected, and maybe we should be studying him. In the absence of being able to study Keith Richards directly, one of the best ways we've been able to experimentally delay aging and improve age-related diseases is calorie restriction. So just feeding laboratory animals less total calories. And they do much better, and I can show the different survival curves, but I always like this picture from the original study, which shows the animals aren't just failing to die and getting decrepit. These animals are the same age. This is the oldest control animal over here on the left, and on the right is a calorie-restricted animal that's the same age. And so there really is an opportunity here to intervene in something fundamental in the aging process and get into a physical condition that I think most people would choose given the option. And so a lot of the work in my lab and in the aging field over the past decades has been guided by trying to understand some of the changes that happen in calorie restriction and whether aspects of that can be replicated in the absence of calorie restriction, in addition to many people experimenting on themselves with actual calorie restriction, which so far has not panned out too well. So I don't want to suggest that NAD, which is what we're working on now, is the whole explanation for this, or even close. There's many hundreds of things documented to change during calorie restriction. There's other things even my lab is working on that change in calorie restriction. But one of the things that really drew our attention to NAD is, sorry if that's changing on me, is it is an example of something that really does change in an age-related way in many mammalian tissues, and that is restored, at least in some tissues, by calorie restriction. So these are NAD levels in skeletal muscles from mice. On the left side, showing ages of 6, 22, or 30 months. So 30 months is really close to the end of lifespan, longer than most mice actually live. And you can see in the muscle, the NAD levels have fallen. On the right is skeletal muscle from calorie-restricted old animals versus controls. And you can see that calorie restriction, in addition to making them live longer and look better, is restoring NAD levels. And so this is NAD, which hopefully all of you remember the molecule I'm talking about, nicotinamide adenine dinucleotide. It's a fundamental redox cofactor that you hopefully remember from biochemistry textbooks, and probably related to these reactions shown in the cycle on the right here. Things like glycolysis, beta oxidation, TCA cycle, take oxidized NAD and reduce it to NADH by accepting high energy electrons. It donates those back to reactions like lactate dehydrogenase that need reducing power, or complex 1 of the electron transport chain. And so this is really fundamental to life. There's no sustainable way to generate ATP without NAD. It's having a severe nutritional deficiency is the cause of the disease pellagra, which becomes fatal if not treated. So we know it's lethal to not have NAD. It's lethal even at the cellular level if you deplete NAD fully in tissue culture. And we think that's really due to the right-hand side of this graph. On the left-hand side, there are enzymes that we've discovered more recently that break down the chemical backbone of NAD and release nicotinamide. So these are things like PARP, CD38, and sirtuins. And that necessitates an NAD salvage pathway to regenerate the NAD pool. And so there's, I think, fundamental roles for some of these enzymes in the first place on the left side that are important in their own right for their activities. But having this system also creates the possibility that you get these things out of balance on the left. And so you deplete the overall NAD pool and deny the cell NAD available for the redox reactions on the right as well. And I think that's when you really get into severe problems. So the loss of NAD is conserved in humans. There's a lot less data in humans, but these are 4 examples of different human tissues where there's been documented to be an age-related decline in NAD. And one nice thing about the system is there are supplements available. So we have the possibility of boosting NAD levels, and our lab and a lot of others now have been focused on this question for a while of will things get better if we just supplement NAD levels? So the answer in rodent models with really high doses seems to be mostly yes in many different conditions. So these are just 3 examples. These are three of the first things that were shown and some of the ones that are guiding human clinical trials. So I tend to focus on these for my example slide, but there's improvements in diabetic phenotypes and glucose homeostasis on the left, Many examples now of improvements in heart failure models. And there's an example here in the middle. And improvements in cognitive function, particularly in Alzheimer's disease models. And so there's an example of that on the right-hand side. But I wanted to show it's not restricted to those phenotypes either. There's really a wide range of things, like too many to list, that have been shown, and this includes improvements in eye health. So I thought for this audience, I would include this example of protecting the retina against light-induced degeneration. So the bottom layer here, so this is the control on top, this is the damaged layer in the middle, and this is nicotinamide riboside-treated animals. So that is one of the NAD precursors that has become popular to study, and you can see there's a very dramatic protection in this case against light-induced degeneration. Our own Ken Schindler here at Penn has done a lot of work establishing the protective effect of SIRT1 in retinal ganglion cells. And so one example from his lab shown here on the right, and that is one of the classic NAD-dependent enzymes. And so I guess it remains to be seen how critical NAD levels are in some of these phenotypes or in enabling some of these phenotypes, but certainly that's an NAD-dependent pathway that seems to be playing a very protective role. And so this is kind of where we are in the field right now. We've got a lot of very convincing evidence in preclinical models, and again, with very high doses. The human trials that are coming out now are more mixed, particularly for things like diabetic phenotypes. It hasn't been very promising. We haven't seen a recapitulation of what's happening in the mice. Other things like mitochondrial myopathies and peripheral artery disease, we are seeing promising effects. And so there's, I think, going to be a use for these things in humans, but there's really a big question mark on why we're failing to see some of these very specific benefits translate in a straightforward way. As we see things fail to translate effectively, it's really shining a spotlight on the fact that we don't really know which of the downstream reactions are critical here. In most cases, we're just supplementing whatever's wrong with high levels of precursors and hoping it gets better. NAD's been documented to work in about 500 different known reactions, so it's involved in just about every pathway at some step. And in almost every case, it's completely unclear where these benefits are coming from in terms of the downstream effects. In addition, it's unclear in most cases which cell type's actually mediating these effects. And NAD is compartmentalized within cells, and so the subcellular compartment is really unclear in most cases. And so a lot of what my lab's focused on now is documenting which of these downstream pathways actually matter, is doing tissue-specific models to try to understand which is the relevant cell type for some of the benefits that we can see. And the example I'll show you about today, I'll just present one little vignette, which is on subcellular compartmentalization. And I hope that maybe this is something where we might find an opportunity for collaboration with some of the people in this room, because I think many of these pathways are conserved across most of the cell types in the body and are certainly likely to be relevant in the eye, as well as in the tissues we're studying. And so the focus for the last few slides is going to be on how mitochondria get NAD. And at the time we started thinking about this, we assumed we were just looking it up in the textbook. But it turned out there was a very small number of labs viciously fighting about these first two models, whether there is a complete NAD synthesis pathway inside mitochondria and they make their own, or whether this intermediate nicotinamide mononucleotide is taken up and the mitochondria complete the synthesis to make the NAD pool. It was known that the model on the right is correct in yeast and plants, but no mammalian homologue of the NAD transporter had been found, and so that model had initially been rejected. And we were stuck with these other two. We were able to do some heavy isotope tracing where we used a strategy that generated labeled NAD only in the cytosol and then could detect that inside the mitochondria. And so to our satisfaction, we're able to establish that there really must be direct transport, that must be this third model that's correct. So we published that in 2018. A couple of years later, we and two other groups identified the transporter as SLC25A51. Which was previously annotated as a mitochondrial transporter, but had unknown substrates, and showed up in all the CRISPR screens people were doing for lethal genes. And so it was clearly flagged as important, but nobody had really put a function on it prior to this. And so this is one of the proof of principle experiments, looking at cells that are knockout for this transporter. If you look at total cellular NAD levels on the left, they're normal. And if you isolate mitochondria, the NAD levels are really low. And one of the expected phenotypes that we thought we would see here proved to be the most dramatic thing that was different about these cells, which was a complete lack of mitochondrial respiration. So this is data from a seahorse, and if you're used to looking at oxygen consumption data from this machine, you'll recognize the pattern. shown here in the dark curve of different inhibitors and activators causing different degrees of respiration. But the important point here is that in the knockout, the line's just flat. There's no real measurable respiration at all from these cells without the NAD transporter. There we go. And so all three of those papers, ours and the other two, showed different cell lines where that had been tested in and seemed to be uniformly true. What hasn't been really established yet is whether this transporter really is the major mechanism in vivo. And so that's what we've been working on since. This is one of the first experiments we did, putting this transporter into an adeno-associated virus and infecting mice and looking at NAD levels in the mitochondria in their livers. And so that's what's shown on the left side here, is that with these AAV transfected, overexpressing this transporter, you do get a nice increase in mitochondrial NAD. There's also an increase in NADH, because the question always becomes whether you caused a redox shift and just oxidized your NADH to get more NAD, but we see more of both. So the pool size is definitely bigger after overexpressing this virus. And the mice are actually relatively normal. I just put this blood glucose graph up here because we consistently see that across every phenotype we do. Fasting, feeding, glucose is always a little bit lower in these mice. But they're otherwise relatively healthy. They're normal size and they behave normally, and nothing really stood out, which was an advantage for being able to put them through some of the next steps that we wanted to take them through. So we decided we needed to apply a stress, and we specifically wanted to now use the model to ask this question of whether shifting NAD into mitochondria is what really is protective when you do supplements. Is it the mitochondrial pool that really matters? So to test that, the model system we decided to use was liver regeneration, because it had been known since the 70s that the regenerating liver gets low on NAD, and that becomes a limiting factor. And so we had shown, and others have shown now, that if you supplement NAD precursors in this model of 2/3 partial hepatectomy, and then wait 48 hours, you see that the ones that got supplemental NAD regrow the liver faster, shown here on the right. And if you look histologically, the liver looks almost normal again with the NR treatment, which is boosting NAD levels. And at this time point, the control livers still have this speckled pattern of fat droplets that's characteristic of liver regeneration. So they haven't really gotten as far along in the process at this point. And so we thought this would be an ideal model to test the effect of shifting NAD into the mitochondria, not just because we knew that the NAD precursors worked so well, but also because we had done this experiment looking about 24 hours after the partial hepatectomy and seen that the natural response to the liver to regeneration is to shift some of the NAD into mitochondria. And so this is the result that was known here on the left, that if you look at the whole tissue NAD level, mid-regeneration, NAD levels fall by about half. And so we recapitulated that. If you isolate mitochondria at the same time point, it turns out that the NAD levels inside mitochondria are actually even higher, despite the falling NAD levels. And we've actually seen this in a few other models now where animals are highly stressed, particularly in hemorrhagic shock models. It seems multiple tissues actually translocate whatever NAD remains into the mitochondria, even as tissue levels are falling. And so we thought this was an attractive system to test the idea that just shifting the NAD into the mitochondria might be protective in the first place. So here's the experimental design. We used that same AAV system, overexpressed the transporter in the mice, and so we're not adding any NAD to the system now, we're just shifting it into the mitochondria. Did the partial hepatectomy, and again waited 48 hours. And this is just showing you that the NAD levels and NADH levels remain high throughout the regeneration process. And if we look at mitochondrial respiration in mitochondria isolated from these livers, you can actually see even pre-partial hepatectomy, there's a little bit of improvement in respiration if you use complex 1 dependent substrates, so NAD dependent respiration. But if you look post-hepatectomy, there's a pretty dramatic drop-off of the respiratory capacity of the mitochondria from the animals that are wild type and undergoing this partial hepatectomy. And having more NAD present in those mitochondria almost completely protects the animals overexpressing from that. That's also reflected in ATP levels, which are maintained much better in the animals overexpressing the transporter. And so here's the final outcome from this experiment. It looks basically indistinguishable from supplementation. So the degree of increased liver growth, liver to body weight ratio here is about the same as we see with nicotinamide riboside. And if you look at hepatocyte proliferation here as a mitotic index, that's also improved substantially with just overexpression of the transporter. And even histologically, this is almost indistinguishable from the effect of supplementation, where you see those lipid droplets clear out of the hepatocytes much sooner, in addition to more replication. And so now with this project, we're at the stage I complained about at the beginning, where there's these 500 NAD-dependent reactions we have to try to assign potential mechanisms to, and we're just getting started on that. But I just thought I would show you some of the metabolite data that we've got. Sorry, I don't know how to back that up. Just to make the general point that if you look here at total metabolites, they're going every which way during regeneration. If you look at mitochondrial metabolites, there is a general depletion in the control animals during regeneration. A lot of mitochondrial metabolites go down, very few go up. And here, if we've supplemented NAD levels in the mitochondria in this overexpression model, you can see you sort of restore a lot of those metabolites. You again have about equal numbers going up and down. And so I think the answer here is going to be very complicated in that NAD is involved in so many of these different pathways where metabolites are fluxing through mitochondria, and that there's a sort of a general inability to keep biosynthesis going full speed without enough NAD in the mitochondria. But we are still really trying to narrow this down to specific pathways and understand exactly which enzymes are responsive to changes in NAD levels. And so with that, I will just sum up by saying NAD levels do decrease with age, even in humans in some tissues. And at least in rodents, we see clear effects, beneficial effects of supplementation. So I hope we'll be able to continue to develop this as a human therapy for different conditions, but needing to put some time into understanding which ones are really going to be responsive. I hope I've convinced you that SLC25A51 is the major determinant of mitochondrial NAD levels, even in vivo from the data we have so far, not just in cell culture. And that at least in this example system that we've really used here, we think that it really is the mitochondrial NAD pool that matters. And so when it comes to supplementation, the reason we might need to use massive doses to get effects in rodents is because that only has a small effect on mitochondrial NAD levels. And so if we can really do something more targeted, that may allow us to take reasonable doses or other pharmacological approaches to activate this transport and really gain some of those beneficial effects that are seen in rodents and not yet in humans. So with that, I will just point out that Shamasta Mukherjee is our mouse surgeon, and the only reason we're able to do these partial hepatectomy models. And Tony Davila and Tim Luongo really pioneered the studies of the transporter in the lab. So I want to thank each of them, and I'm happy to take questions.
Joe, great talk. So how does caloric restriction affect NAD levels?
That is a great question. We don't know. I mean, it does induce expression of the key biosynthetic enzyme NAMPT, nicotinamide phosphorybosyltransferase. And that is downstream of AMPK, which is a key energy sensor in the cell. So there's this idea that just having low energy sort of automatically triggers that signaling pathway that upregulates NAD synthesis as a general compensation. But that's not, we don't have knockout data to prove that that's essentially the mechanism that's taking place.
I don't mean to monopolize questions if someone else has one. So it turns out that mutations in NMNAT1, which is the nuclear isoform of NMNAT and required for NAD synthesis, cause non-syndromic retinal degeneration, very severe retinal degeneration. And it looks like in animal models of that disease that the degeneration occurs because of failure of DNA repair mechanisms to repair oxidative DNA damage that occurs just due to baseline metabolism. So you're considering DNA repair processes as one of the 500 downstream NAD systems that you're looking at for what matters?
Yeah, I mean, for sure, PARPs, right? Polyadipobasyltransferases are NAD-dependent enzymes, and that's certainly one of the proximal targets when you have low NAD levels, is if you can't pyrrolate things that you lose a key signaling mechanism related to DNA repair. Another one that comes up a lot is PGC1 alpha is downstream of SIRT1. And so that controls expression of many of the key antioxidant enzymes and some of the repair enzymes. And so that just lacking PGC1 alpha activity can also make the cell much more susceptible to DNA damage.
Maybe not. Great talk. So you showed nicely in the liver that the mitochondrial NAD seems to be more important when you put your transporter in. Is there any evidence from other tissues that it behaves the same way, or presumably there's some role for having the NAD outside the mitochondria as well?
Right. Yeah, and certainly, if you fully deplete NAD, you cause all kinds of chaos in any cell type or any tissue. But there is now genetic evidence linking aortic aneurysms to mutations in SLC25A51, and also to other NAD biosynthetic enzymes, but the signal's much stronger for A51, and so the implication is that it really is the mitochondrial pool there. And we are in the middle of doing some studies now in heart failure, where it does seem to, again, be the key transporter, and we're not, I guess the N is probably too low to make any claims at this point, but certainly we and a few other groups are working actively on this.